Okay, you guys, in this video, we're going to go over topic 3 of the 5.1 notes. In topic 3, we're going to talk about a lot of basics regarding genetics and inheritance, and how traits are passed on from parents and their offspring. And to do that, we're going to talk about a lot of this stuff in the context of Gregor Mendel, who was this Austrian monk in the 1800s who figured out a lot of the basics that explain how traits are passed on from parents to their offspring. And so this guy, Gregor Mendel, like I said, he was a monk, and this was in the second half of the 1800s, so about 150 years ago. This guy was doing a lot of crosses between pea plants, producing offspring from different pea plants, and he did hundreds and hundreds of crosses, making thousands and thousands of offspring. And what he did, he basically just looked at different traits that the parent plants had, and then when he crossed them and produced offspring, he looked at how those traits were passed on in the offspring and what traits the offspring had. And he started noticing a lot of predictable patterns that kept repeating when he was doing his experiments. And from those patterns, he came up with some concepts that he believed explained what was going on in these crosses and in these traits that he was tracking in the pea plants. And it turns out a lot of those are completely accurate and explain a lot of how genetics and how traits are passed on between parents to their offspring. And so for that reason, he's known as the father of genetics. It's a lot of these things that we're going to talk about. It's pretty impressive that he figured out, because this was before they knew anything about DNA. They didn't know it existed yet. Chromosomes, we didn't know those existed yet in the 1860s, 150 years ago when he was doing these experiments. And what's cool is that as we started to learn and discover in science about DNA and of chromosomes and meiosis, producing gametes or sex cells that are then going to be physically used to produce your offspring, all of it really kind of meshes very well with these ideas that he came up with. And we found out that there's actually some very accurate molecular explanations for these patterns that he was observing. And so I'm going to kind of go over some of the gist of his experiments and what he observed, and then we'll go over some of the basic concepts that he figured out, and talk about how, with the knowledge we know today about biology and DNA and chromosomes, how what he figured out is very consistent with all the stuff that we know today. And so in his experiments, a lot of times he was starting with these true breeding pea plants. And so a true breeding plant is a plant that when it's self-pollinated, it always produces a certain trait in the offspring. And so plants are pretty cool because for a lot of flowering plants, they can pollinate themselves and produce seeds and offspring with just the one organism. And so a true breeding plant would be, for example, if we had a true breeding purple flowered plant. So we have a pea plant who has purple flowers, and if we say it's a true breeding purple flowered plant, that means that this purple flowered pea plant always reliably all the time produces offspring that have purple flowers, like it has when it's self-pollinated. So when that flower or that plant is self-pollinated and produces seeds, those seeds always grow up to have purple flowers as well. That's what a true breeding plant is. And so in his experiment, what he would do is he would take two true breeding plants that had opposing characteristics for a certain trait, or opposing appearances for a certain trait, and then he would cross them to produce what we call hybrids, which are the offspring that result from these two opposing true breeding plants. And then he would take those hybrid offspring and then cross them with themselves to produce a second generation then of pea plants. And so, for example, he would take a true breeding purple flower plant that always produces purple flowers, and he would take a true breeding white flowered plant and he would cross them together. And that would produce what we call a hybrid in this context, which is a plant that results from two different true breeding plants, opposing true breeding plants. And that would be what's called the F1 generation. And so the two original plants, we call that the P generation. And then when they produce offspring, we call it the F1 generation. And then what he did is he took those F1 plants, the plants in the F1 generation, the hybrids, and he would cross them with themselves or self-pollinate them, and produce what's called the F2 generation, which is the third generation here, the grandkids. And then he looked at how that trait, in this case flower color, was passed on. And so here's what his observations were. When he took true breeding plants and crossed them with each other, in the F1 generation, the hybrids, they all had just one of the traits that the parents had. And so, for example, with the purple flowers and the white flowers, he crossed them. Those two true breeding parents, and 100% of the offspring had purple flowers. Like every single pea plant that was produced from this cross always had purple flowers. And he produced hundreds and hundreds of these purple flowered plants. All of them always had purple. And then what he took these hybrids who had purple flowers, and he crossed them with themselves or with other hybrids in the F1 generation, other purple flowers in the F1 generation. And when he crossed them in the next generation, he observed that there was a lot that still had purple flowers, but there were some that then also had white flowers. And he also noticed that there was this this proportion that kept popping up in the F2 generation, where it was a 3-to-1 ratio, or 75% had the purple flowers, or 25% had the white flowers on average. Which, if you think about this is kind of a strange result to get if you don't really know anything about genetics yet. Like if you take a purple flower and a white flower, they produce all purple flowers. But then for some reason, if you take those purple flowers and you cross them with other purple flowers in that generation, all of a sudden now there's flowers that have white flowers. It's kind of weird. And he did that for lots of different traits, not just flower color, but he did it for seed color and seed shape and pod color, flower positions, the height of the flower, for lots of different traits. And again, he had the same basic setup where he would take true breeding parents that had opposing characteristics, and then produce the hybrids, and then take the hybrids and then cross the hybrids with themselves to produce the F2 generation. And in the F2 generation, he kept getting this 3-to-1 ratio for these traits that he was tracking. And so with this information, these observations, he came up with an explanation for why this might be happening. And so there's these concepts that he comes up with involving genetics and inheritance. And so that's what I'm going to talk about right now. I'll kind of explain some of those concepts right now, and then kind of show you how today, with what we know about DNA and chromosomes, how it's very consistent with what he figured out, which is pretty cool. So, for example, one concept he came up with, which this is kind of an obvious one, is that there's different versions of traits that exist. So for a given trait, there's going to be different versions that exist. So for flower color, there's different versions of flower color that exist. There's purple and there's white. For seed color, there's different versions of seed color. You could have green seeds, you could have yellow seeds. So first and foremost, he figures out that, okay, yeah, for certain traits, there's going to be different versions that exist. And so those are called alleles. Soon we'll start calling those, or soon in the course of science, we start calling those alleles. Alleles are the different versions of traits that exist. So there's purple alleles that code for purple flowers, and there's white alleles that code for white flowers. And so there's that. And then he figured out, or not figured out, but his second idea was that for each trait or each characteristic, you get 2 alleles. Inside an organism, you're going to have 2 alleles for each trait. And so there's these different alleles that exist, and for each trait, you're going to have two alleles inside of you somewhere. He doesn't know what it is, but something inside of you. There's 2 sets of instructions you have for each trait or characteristic. So for flower color, you have 2 sets of instructions inside of you for flower color, or 2 alleles. Maybe you have instructions for purple. You have a purple allele, and you have instructions for purple. Maybe you have 2 sets of instructions to have purple flowers. Or maybe you have instructions for purple flowers and instructions for white flowers. Or maybe you have instructions for white flowers and instructions for white flowers. But you have 2. You have 2 sets of instructions inside of you for each trait or characteristic. Which again, today we know that to be very true, and we'll talk about that in a second. And then his third concept was that if the 2 alleles that you have for a certain trait are different, which happens sometimes, maybe you have the instructions for purple flowers, but also inside of you have the instructions for white flowers. That happens. And what he suggested is that, or figured out, is that certain alleles are going to be dominant over other alleles. And so when you end up with a situation where you have 2 different sets of instructions for a certain trait, that one of those alleles is going to win. And so that when you look at the appearance of that organism, you're only going to see the evidence of one of those alleles inside of that organism. And then the other one is going to be masked or hidden, and you're not going to really see that in the physical appearance of the plant. And that would be the recessive one. So for flower color, for example, if you have a flower who has, again, 2 alleles, 2 sets of instructions for flower color, and one set of instructions says purple flowers, and one set of instructions says white flowers, well, then one of those is going to win. And in the case of pea plants, it's purple. Purple is going to be dominant. So that plant would have purple flowers, and it would just be just as purple as all the other flowers that are purple, even though it technically has instructions for white flowers as well. But that's going to be completely hidden and masked in the appearance of the plant. And we would call the white allele the recessive allele, and then the purple allele the dominant one for flower color in these pea plants. And then another concept, one last concept I'll explain right now, is he came up or discovered that when you produce offspring, when a pea plant is going to produce offspring, that pea plant can only pass on one allele that they have for each trait or characteristic. And so the pea plant would have 2, right? You have 2 alleles for every trait or characteristic. But when you have an offspring, you can only pass on one of those 2 to your future kid. So when you produce some kind of packaged gamete, some kind of sex cell that's going to be passing on the information to your kid, and that cell that you're going to pass on to produce your kid is going to be the instructions, is only going to be one of the sets of instructions you have for each gene, not both. So for example, if you had, again, if you were a pea plant and you had 2 alleles for flower color, one of them was purple and one of them was white, you have a purple allele and a white allele, instructions to have purple flowers, instructions to have white flowers. When you have kids, you can only pass on to your kid or your future offspring, you'd only pass on to your kid one of those alleles. So you can either pass on the purple allele or you can pass on the white allele, but you can't pass on both of them. You, as one of the parents, can only pass on one of them. And then the other parent is going to have to pass on the other allele. But you only pass on one, not both, for each gene or characteristic or trait. And so that's called the law of segregation, the fact that you have 2 alleles for each trait, but you can only pass on one of them to your kids. And so now I kind of want to go back and explain on a biological level, with information that we know today, how this is all very consistent with stuff we know about DNA and chromosomes and meiosis. And so, for example, this idea that there's different versions of traits that exist or characteristics that exist in an organism is explained today by the fact that your genes are sequences of DNA that code for specific proteins that are going to produce specific traits that you have. And that DNA sequence can be slightly different and code for a slightly different version or functional or non-functional form of a protein to produce a different trait. And so, for example, I'll keep using the example of flower color. For flower color, like I said, there's a purple allele and a white allele. But what those alleles actually are, what we know today is that those alleles are actually a sequence of DNA located on a chromosome that you have somewhere that has the instructions to produce a protein, usually, that's going to then produce that flower color. And so if you have purple flowers, it's because there's a sequence of DNA that you have at a special location on your chromosome that codes for a protein that produces purple flowers. But then you could have different versions of that exist. Like there's other alleles that exist, like for white flowers, there's an allele for white flower that exists, and that allele is actually also a DNA sequence that you would have at the same location on the same type of chromosome. But that DNA sequence would vary a little bit. It would be a slightly different sequence of DNA that leads to a different protein or a different functional variant of that protein, maybe a non-functional variant of that protein. So in this case, for flower colors, if you have this sequence of DNA, it leads to no functional enzyme being produced, and so you're not producing these purple pigments. So then you would have white flowers, for example. And so different alleles are actually different DNA sequences that you could have on your chromosomes. And then you're going to get two of those because you are going to get a set of genes and a set of chromosomes from your mom, and you're going to get a set of chromosomes and a set of genes from your dad. So you have homologous chromosomes, which we've talked about in the previous videos in this unit, where your mom and your dad each gave you a chromosome with the same set of genes on it. And so in this case, for pea plants, the plant is going to have a gene for flower color, but it's going to have 2 copies of that gene because it's going to have a chromosome from the dad plant that it got with instructions for flower color, and it's going to have a chromosome from the mom plant that it got with instructions for flower color. So you will end up with 2 alleles for a trait because you have homologous chromosomes. Each parent is giving you a set of chromosomes, and you're a diploid organism, which for humans and a lot of other animals and certain plants like pea plants, they are diploid organisms, and they're going to get a set of chromosomes from the mom and a set of chromosomes from the dad, which are then going to give you 2 alleles for each gene. For any given gene, whether it's seed color, flower color, seed shape, you're going to have 2 sets of instructions, one on a chromosome that your mom gave you and one on the chromosome that your dad gave you. So that's super consistent with what Mendel was thinking. And then some of those alleles are going to be dominant sometimes over the other alleles, like for this case with flower color, because one of these alleles, for the allele for purple flower, is a specific sequence that produces this functional protein that produces purple pigment. And this allele for white flowers is a different sequence of DNA that doesn't produce a functional enzyme, producing no purple pigment. That means that if you ended up with both, because you're going to get 2, and let's say you got on one chromosome from one parent, you got the DNA sequence that leads to this protein that makes purple pigment, but on the other allele you got from the other parent, it has this DNA sequence that doesn't lead to a functional protein. Well then, since this guy is not making any pigments, but you do have this sequence leading to this protein that is making purple pigments, it's maybe not too surprising that, yeah, you're going to have purple flowers still, because you still have the instructions on one of those alleles. There is the instructions to make that protein that makes purple pigments. So you're going to have purple. So we would say purple is dominant because even if you end up with purple and white, the white one's not really doing anything. And the purple one is still having the instructions to make that enzyme that makes purple pigment in the flowers. And so purple would win. If it was between purple and white, you're going to end up with purple flowers because white's not making anything and purple is making something. And if you end up with both, well, then you're going to be making the purple pigments. And so there are a lot of situations where you are going to have a dominant allele over a recessive allele just because of the way those proteins get produced from those different DNA sequences. One of them might be a winner, and one of them would be completely invisible because of the other allele being present. And then for those alleles, we typically represent the alleles by a capital or lowercase letter. So a dominant allele is usually represented by a capital letter, and a recessive allele is represented by a lowercase letter. And for a specific trait, we usually use the same letter for a specific trait. So first, talking about flower color, and there's different alleles that exist for flower color, like purple and white, we would try to use the same letter. So if you're talking about the same gene, like the gene for flower color, we would use the same letter to represent the dominant and recessive allele. We would just use a capital and lowercase version of that letter for each allele, depending on which one's dominant, which one's recessive. And usually, a lot of times we use the allele that's dominant, we use that letter. So in the case of flower color, purple is dominant, so we use the letter P. And so we'd use an uppercase P to represent a purple allele and a lowercase p to represent a white allele, since it's the recessive one. So then I want to just go over some vocab with you guys, some basic genetic vocab. There's something called genotype. Genotype is a word we use today to refer to what are the 2 alleles you have for a certain gene. So for every gene, you're going to have 2 alleles, just like we said. So like for flower color, the flower color gene, a pea plant is going to have 2 alleles. The 2 alleles that it has, that's the genotype. So you could have 2 purple alleles, like a big P and a big P. That could be your genotype. You have a purple allele, and then you have a purple allele. You have the instructions to have purple flowers and the instructions to have purple flowers. That would be your genotype. Or maybe you have 2 white alleles. You have 2 of the recessive alleles. You have instructions to have white flowers, instructions to have white flowers. That would be your genotype. Or you could you could have one of each. You could have instructions to have purple flowers and instructions to have white flowers. So maybe you have a dominant allele and a recessive allele. But that's going to be your genotype. Now, if those 2 alleles are the same, we say that your genotype is homozygous. Like if you have 2 purple alleles, we would say that you're homozygous. Or 2 recessive alleles, we would say you're homozygous. Either homozygous dominant, you have 2 of the dominant alleles, or homozygous recessive, you have 2 of the recessive alleles. Now, if you have 2 alleles that are different for a gene, which happens a lot, maybe the 2 alleles you have don't match, we say that you're heterozygous. So that's what genotype is. And then phenotype is your actual physical appearance that you're going to express because of those two alleles that you have. So you have these two alleles, but then ultimately you're going to have some kind of physical expression in the organism of that information. And that physical appearance is called your phenotype. So for flower color, I mean, you're going to either end up with purple flowers or white flowers. That's going to be your phenotype. And it depends on the two alleles that you have. If you have two purple alleles, well, then yeah, you're going to have purple flowers. Or if you have two white alleles, well, then yeah, physically you're going to have white flowers. And if you're heterozygous, if you have a purple and a white allele, because purple is dominant, you're going to have purple flowers. That would be your phenotype. So this would lead to purple, that would lead to purple, this would lead to white. So think phenotype as like the pH, and phenotype is like the pH and physical appearance, or like the physical expression of that gene that you're actually going to see visibly in the organism. And then another explanation for this law of segregation, a biological explanation for this law of segregation, this other concept that Mendel came up with, the fact that you can only give one of the alleles that you have. So whether you have two purple alleles or a purple and a white allele or two white alleles, you can only pass on one of them. We know that is true today because through the process of meiosis is what's going to take a cell and produce gametes, your sex cells, which are like sperm and egg cells. And in those sperm and egg cells, those gametes, you're only going to have one of each type of chromosome in those sex cells. So your homologous chromosomes during meiosis are going to separate and be divided into the final daughter cells, each carrying only one set of instructions, only one set of alleles for each trait that you have, or each gene. So in this example, let's say if you look at this picture, again, let's say we're talking about pea plants. And so for pea plants, this is looking at the R's is for this gene for the seed shape. You could have round seeds or you could have wrinkled seeds, and round is dominant. But let's say you're heterozygous. You have instructions to have round seeds and you have instructions to have wrinkled seeds because you have a chromosome from mom and you have a chromosome from dad, right? You have two alleles. But when you have kids, your cells are going to have to undergo meiosis. And when they undergo meiosis, as you can see in this diagram, you're going to end up with these daughter cells that only have one of those chromosomes in the homologous pair. So you're either going to have a chromosome with the instructions for round seeds like these guys, or you're going to have a chromosome in your egg cell or sperm cell with the instructions for wrinkled seeds. But you're not going to end up with an egg cell or sperm cell at the end of meiosis that has both alleles. Like you'd only pass on one of them. And so that's the law of segregation. You have two alleles, but in the end, when you produce sex cells that are going to be passed on to your offspring, there's only going to be one of those packaged into your sex cells. And so moving on, then we can talk about Punnett squares, which kind of allow you to see the different combinations in which two parents could pass on alleles to their kids, and the possible ways that the potential offspring could inherit one allele from each parent. And so what you do on a Punnett square is you basically put the possible alleles that one of the parent can pass on goes on the left side of the Punnett square, and the possible alleles that the other parent could pass on would go on the top side of the Punnett square. And then inside the Punnett square, you would join those alleles together. So like you could have this allele. So let's look at an example here. Let's say you're taking like those F1 plants from Mendel's experiment, the hybrids that, well, let's recap actually a little bit more. Let's look at this whole picture here. So just to go back to Mendel's experiment that I explained at the beginning, let's say you had a true breeding purple flower plant and a true breeding white flower plant. Well, the reason they're true breeding, what we know, what Mendel figured out is true breeding means that you just have two alleles that are the same. And so this purple flower plant has two alleles for flower color and they're both purple. It's homozygous. And this true breeding white flower plant is homozygous recessive. So that's ultimately what it means to be true breeding. It just means that you're homozygous. And so anyway, when you cross them, this parent can only pass on a purple allele, right? You have two purple alleles, you got to pass on one of them. And this parent has two white alleles, or two recessive alleles. And so this parent's going to have to pass on a white allele, and only one of them. And so all of the F1 kids, every single time, it makes sense now, are going to always get a purple allele from this parent and a white allele from the other parent, because they can each only give one and pass on one according to the law of segregation. So these kids in the F1 generation are always going to inherit a dominant and a recessive allele, one from each parent, which would make them heterozygous. So in the F1 generation, they're all going to be heterozygous and have an allele for purple and an allele for white, which means they should all be purple because purple is dominant. And that's exactly what happened. They all 100% were purple. Now, when you take these F1 plants and you cross them with other F1 plants, so these purple flowered plants in the F1 generation, when you cross them with each other, well, then you're taking two heterozygous plants and crossing them. So you're going to have one of the parents is heterozygous, and the other parent is also just like that parent. It's also heterozygous. So you're taking a plant that's big P, little p, and you're crossing it with another plant that's big P, little p. Well, in that situation, we get set up a Punnett square to show what kind of kids are possible from that, because one of those parents who's big P, little p, they could either pass on a purple allele, a dominant purple allele, or they could pass on a recessive white allele, right? You have one of each. And so when this plant does meiosis, the cells in this plant, when it does meiosis, half of its sex cells at the end of meiosis will end up with the purple allele, and half of them will end up with the white allele. So it's going to be one or the other, right? And then the other parent, same thing. The other parent's also heterozygous. So, and the other parent, who also has a purple and a white allele, half of that parent's sex cells at the end of meiosis will have the purple allele, and half of them will have the white allele, because you can only pass on one, not both. And so on a Punnett square, on the left side of the Punnett square, we would put the types of sex cells, the types of gametes, or in this case, the type of eggs from the mom that are being produced in the plants, which some of those eggs will have the purple allele, some of them will have the white allele, but not both. And then same thing for the dad. The sperm that's being made by the dad will either have a purple allele or a white allele, not both. Some of the sperm has purple, some of the sperm has the instructions for white alleles. And so in the Punnett square, you could see the combinations that sperm and egg could meet. So maybe it's this sperm from dad carrying purple and this egg from mom carrying a purple allele. So then you're going to end up with a kid who got two purple alleles, one from dad's sperm and one from mom's egg. Or you could have a kid like this who got this type of sperm from dad that's carrying the purple allele, and this type of egg from mom that's carrying the white allele, and then you end up with this kid right here who's now heterozygous. It got a purple and a white allele, one from mom, one from dad. And so the Punnett square is going to show you the different ways that mom and dad could each give one allele to the kid. So really, here are all the possible ways that could play out. If it's this sperm and this egg, or this sperm and that egg, or this egg and that sperm, or this egg and that sperm. And here are the different alleles, the way they could combine in the offspring. And then, so this Punnett square, the results of this Punnett square will tell you the expected genotypic and phenotypic ratios in the offspring. And so you can see in terms of genotypes, you have some kids who are homozygous dominant, you have some kids who are heterozygous, you have some kids that are possibly going to be homozygous recessive. And so when I look at the genotypic ratio right here, I could write it like this. I could say in the offspring from this cross, for every one kid that has two purple alleles, there should be two kids that are heterozygous and one kid that's homozygous recessive. So there's a 1 to 2 to 1 ratio in terms of the genotypes that I should expect to see in the offspring. And so I don't know how many kids they're going to have. This doesn't mean that they're going to have 4 kids. This is just showing you the 4 ways that egg and sperm could combine. But once they do have kids, maybe they have thousands of kids. Among those kids, there should be about a 1 to 2 to 1 ratio in terms of these genotypes that the kids have, or 25%, 50%, 25%. 25% of the kids should have these 2 alleles, 50% should have those 2 alleles, and 25% should end up with 2 recessive alleles. And then we could also look at the phenotypes in terms of how they physically look. Well, if you end up with 2 purple ones, you're going to be purple. If you end up heterozygous, you're also going to be purple because purple is dominant. And if you end up with 2 white alleles, well, then you're going to be white. And so we would say that's a 3 to 1 ratio. There's 3 outcomes that lead to being purple for every 1 outcome that leads to being white. So I should see in the offspring a 3 to 1 ratio, or 75% should be purple and 25% should be white, which is exactly what Mendel found out in his experiment in the F2 generation. When he crossed those F1 hybrids with each other, there was a 3 to 1 ratio, or a 75%, 25% outcome in the phenotypes for those kids, which matches perfectly his model here that he created with these ideas of dominant recessive alleles, and you have 2 and you can only pass on 1. It's exactly what's happening here. So then there's something called a test cross that you can do. So like, let's say there's a plant that has the dominant phenotype. If you have a plant that has the dominant phenotype, you don't necessarily know its genotype. And so, for example, if I have a purple flowered pea plant, well, there's 2 reasons you could have purple flowers. One could be maybe you have 2 purple alleles. That's one genotype that would lead to being purple flowered. Or you could be heterozygous, because heterozygous pea plants would also have purple flowers. And technically, you don't know necessarily just by if you looked at a random purple plant, you don't know what 2 alleles it has. It could be homozygous dominant or it could be heterozygous. So to find out, you could do something called a test cross, where you take that dominant phenotype plant that you technically don't necessarily know the genotype of, and you cross it with a recessive plant, a plant that has the recessive phenotype, because a plant that has the recessive phenotype, you actually do know its genotype. If you have the recessive phenotype, you have to have 2 recessive alleles. You have to be homozygous recessive. That's the only thing that would lead to having a recessive trait, a recessive phenotype. And so you could take that dominant purple flower and cross it with a white flower, and you could look at the kids. And by looking at the kids that are produced from that cross, you could figure out if that purple flower was homozygous dominant or heterozygous. So for example, if all of the offspring are purple, you cross these 2 plants and every single offspring is purple. Well, that would only happen if that plant had been homozygous dominant. If that plant was homozygous dominant and had a purple, 2 purple alleles, like this example right here, and the other parent had 2 white alleles, because we know that for sure because it has white flowers, and you cross them, if you look at the Punnett square, well, all the eggs that parent is making, if they were purple, hypothetically, and all of the sperm that the other parent was making are all have the white allele, well, then all of the kids will always end up with a purple and a white allele, one from each parent, which would mean all of them should be purple. So if all the kids are purple, the only way that's possible is if the purple flower was homozygous dominant. That would lead to all the kids being purple. But if you cross them and you have some kids that have white flowers from this cross, well, the only way that some of the kids could have white flowers, that would be a dead giveaway that parent that had purple flowers must have been heterozygous, because the only way you can end up with white flowers from this type of cross is if that parent had a purple and a white allele, and some of them got the white allele from that parent along with the white allele from the other parent, which would lead to some homozygous recessive individuals. So that's what a test cross is. It's basically just taking a plant with a dominant phenotype whose genotype you don't know for sure, and crossing it with a recessive individual, and then look at their kids, and that will give you clues about, okay, what does that parent have to be? What genotype, what 2 alleles does that parent have? It usually can make it very obvious. Okay, I also want to talk about one last thing in this topic about what Mendel did in his experiments. One other thing that he did in his experiments was he would track not just one trait at a time in the parents and the offspring, but he would track multiple traits at the same time. So like I talked about earlier, he was in a lot of his crosses, he was tracking like just one trait. Like he was looking at the flower color of the parents and the flower color of the offspring, or the seed color of the parents and then what seed color did the offspring have. But there was a lot of crosses and experiments that he did where he was tracking multiple traits at the same time. So he was looking, for example, at the flower color and the seed color of the parents, and then looking at the flower color and the seed color of the offspring at the same time. And in those type of crosses, one very popular type of cross that he did was called a dihybrid cross, where he was tracking multiple traits at the same time. Now, a dihybrid is a plant that is heterozygous for 2 different traits. And so for one trait, they're heterozygous, and another trait, they're heterozygous. And then what he would do is take those dihybrids and cross them with each other to produce offspring, and then he looked at how those phenotypes were inherited in the offspring. So here's an example of a dihybrid cross. So we could do a dihybrid cross that contains 2 plants that are heterozygous for seed color and seed shape. So for seed color, yellow is dominant, green is recessive. And for seed shape, which is a different trait, a different gene, there's round seeds and wrinkled seeds, and round seeds is dominant. And so what he would do is he would first create dihybrids by taking true breeding parents. So you guys look at this picture right here. If we had a true breeding parent who had yellow seeds that were round, and so true breeding means, now we know that means that they're homozygous. So they're homozygous dominant for seed color for yellow, and they're homozygous dominant for round seeds. And so this is showing you guys 2 sets of genotypes here, genotypes for seed colors, genotypes for seed shape. And then crossing that with a parent who is homozygous recessive for both those traits. So this parent is homozygous for green seeds that are wrinkled. Anyway, when you cross those 2 true breeding parents in the P generation, the original parents, you're only going to get one outcome, which is going to be these dihybrids, because this parent has to pass on one of these Y alleles and one of these R alleles. So that's the rule. If you guys remember from the law of segregation, you can only pass on one allele for each trait. So for the seed color trait, this parent has to pass on a yellow allele and has to pass on a round allele. That's the only thing that this parent can do. It has to pass on one of these and one of those. And then this parent, same thing, has to pass on a green allele and a wrinkled allele, because it has to pass on one of these green alleles and one of those wrinkled alleles. So this parent will always be passing on in its gametes a dominant Y and a dominant R, and this parent will always be passing on a recessive Y and a recessive R, which means the F1 generation will always get a dominant allele from a dominant Y or yellow allele and a recessive green allele from each of those parents. So that kid is going to be heterozygous for seed color. It's going to get a big Y and a little y, one from each of those parents. And then for seed shape, it's going to get a big R and a little r, one from each parent. And so it'd be heterozygous also for seed shape. So the kids would be heterozygous for both. That's what we call a dihybrid. You're heterozygous for two different traits, which the kids would then look like yellow round seeds because yellow is dominant and round is dominant, just like this parent. So then what he did is he took these dihybrids in the F1 generation and he crossed them with each other. So now he's going to take two of them, this dihybrid, and cross it with another dihybrid to produce offspring. And in the offspring that he produced, which again, he did these crosses many, many times and produced hundreds of thousands of offspring. In the F2 generation, so the offspring that resulted from that dihybrid cross ended up with a very predictable phenotype or a very consistent phenotypic ratio in those resulting offspring, which was a 9 to 3 to 3 to 1 ratio, where where 9 of them, for every 9, there was 9, a majority that had two dominant phenotypes. So they were yellow and round. They had both dominant phenotypes. And then there were some that had a dominant and a recessive phenotype. So like there were some that were recessive for seed color, but dominant for seed shape. So they were green but round. But then there was also some that were yellow and wrinkled. So they were dominant for seed color, but recessive for seed shape. And then there were some that were recessive that were green and wrinkled, which is both the recessive phenotypes. And it came out to a 9 to 3 to 3 to 1 ratio is what this is showing you here. So for every 9 that were dominant, there was 3 that were dominant recessive, and there was 3 that were recessive dominant, and there was 1 that was recessive for both phenotypes in the F2 generation when crossing these dihybrids with each other. And so the results from that can be explained by this law that Mendel came up with called the law of independent assortment, which states that when a parent passes on an allele for a certain trait, it has no effect on what allele it passes on for another trait and the next trait and the next trait. And so to do a cross involving multiple genes, what you have to do first is you first have to figure out, okay, what combinations of alleles can each of these parents pass on? When these parents are making gametes, sperm or egg cells, what combination of letters are they going to put in those sperm and egg cells? Because the rule is, well, first of all, there's the law of segregation. You can only give one allele for each gene. So for the seed color gene, you can only give one Y, one of your alleles. And then for the seed shape gene, you can only give one of your alleles. Even though you have two, you can only give one. So that's the law of segregation. But the law of independent assortment, what Mendel figured out, is that whatever allele you give for seed color should have no effect on what allele you give for seed shape. So like whatever Y you give should have no effect on what R you give in this example. And that all combinations should be equally possible. Like it should be completely random. These alleles should assort independently when they're making gametes, the Y's and the R's. So for example, in parent number 1, who is a dihybrid, they have to give a Y, they have to give an R. Well, they could give a big Y and a big R. That's one thing that they could pass on to their kids. So that's one type of sperm. Let's say this is dad. That's one type, let's say this is mom. That's one type of egg cell that this mom can make. But there's some eggs that this mom is making that have a big Y with a little r. That's also possible. You have to give a Y, you have to give an R. Sure, you can give a big Y with a little r. Or you can give a little Y with a big R, or you could pass on a little Y with a little r. It has to be one of each. But according to the law of independent assortment, all possible outcomes should be equally likely. Like whatever Y you give should have no effect on what R you give, which means all of these gametes that mom is making should be possible and equally possible according to the law of independent assortment. Same thing for the other parent. The other parent's also dihybrid. So if that's dad, he's making 4 types of sperms. A fourth of his sperm have those 2 alleles, a fourth of them have a recessive and dominant one, a fourth of them have a dominant recessive one, and a fourth of them have 2 recessive alleles. So if that's true, and you are these 2 parents are assorting their alleles independently when they're making gametes, creating these different outcomes, well, then there's lots of different ways that different types of eggs could meet different types of sperm to produce offspring. So then in your Punnett square, you would set up a 4x4 Punnett square this time. And in your 4x4 Punnett square, the reason it's 4x4 is because there's 4 types of eggs. There's 4 combinations of alleles that mom could pass on. and there's 4 types of alleles that dad could pass on in the sperm. And so when you set up the 4x4 Punnett square, it'll show you all the different ways that egg and sperm can meet. So it could be this egg and that's sperm. So that means this kid would end up getting a big Y and a big Y for mom and dad, and a big R and a big R for mom and dad. And so when we connect those alleles back together, we put the Ys back together and the Rs back together. When you're showing these possible genotypes in the offspring. Or, like another example, it could have been, let's say this one right here, which is a big Y and a little R, with this sperm that's a big Y, a little R. So this kid would get a big Y and a big Y for mom and dad, and a little R and a little R for mom and dad. So that would be the kid's genotype, big Y, little r, little r. Homozygous dominant for seed color, homozygous recessive for seed shape. Or like this kid would get a big Y from one of the parents and a little Y from the other parent, and a big R from one parent and a little r from another parent. So this kid would be a dihybrid, actually. It would be heterozygous for both traits. And when we put those letters back together, make sure that you guys always put the dominant allele first. So if there's a dominant allele, that one goes first. So you put the alleles back together, the Ys with the Ys, the Rs with the Rs, but if there's a dominant one, that one goes first. So you'd end up with this genotype for this kid right here. And so this is showing you all the different genotypes you'll end up with, and it even shows you in this picture the phenotypes those kids would have depending on those alleles that they have and what's dominant and what's recessive. And this outcome actually shows you that you should get this 9 to 3 to 3 to 1 ratio. For every 9 kids that are dominant, there's 3 kids who are dominant recessive and 3 kids who are recessive dominant and 1 kid who's recessive recessive, which matches perfectly with what happened with Mendel's dihybrid crosses. When he was taking 2 dihybrids and crossed them for 2 different traits, whatever 2 traits they were, whether they were dihybrids for seed color and seed shape, or flower color and flower position, or pod color and pod shape, this is the outcome he got when he crossed dihybrids, which supports his conclusion that there's this law of independent assortment, that these alleles are assorting independently. And so that's what that cross is showing here. That's how we would do a cross involving multiple genes. So whenever you guys are doing a cross involving, like, let's say 2 genes, you first need to figure out, if you want to see all the possible outcomes, you first need to figure out what combinations of alleles could one parent pass on, and what combination of alleles could the other parent pass on, and then set up your Punnett square accordingly depending on how many combinations are possible. In this case, there's 4 combinations and 4 combinations possible, so we have to set up a 4x4 Punnett square. In your guys' notes, there's some practice questions at the end where you guys will do a couple of these crosses involving multiple genes to see if you can figure it out. Now, today, that can be explained, this law of independent assortment can be explained today with our information that we know about chromosomes and DNA and meiosis, how that gametes are made in organisms, by the fact that during meiosis, your homologous chromosomes are pairing up. If you guys recall from topic 1 and 2, the homologous chromosomes pair up during meiosis, and they line up in the middle and then are separated from each other in meiosis 1, and then the sister chromatids are separated in meiosis 2. But the way those homologous pairs line up can be flipped or flopped. It's completely random, the orientation that they're lining up in pairs. So for example, like you could have like these 2 lining up like this and those 2 lining up like this during meiosis 1, but it also could have been flipped a little bit. Like you could have had them line up like this and this, where now you have like the blue one on this side, which is the same side as this red one, and this red one on the same side as this small blue one, which is going to lead to a different combination of alleles being passed on to the gametes. Like in this case, it leads to the big A and the big B ending up together in some of these, and the little a and the little b ending up together in some of these gametes. Whereas if they had lined up differently during meiosis, which is perfectly possible, it's very random how they're lining up, then you would end up with the big A and the little b ending up together, and you would end up with the little a and the big B ending up together in some of these gametes. And you'll see that, yeah, there's 4 possible outcomes here. You could end up with big A's and big B's together, or you could end up with little A's and little B's, or big A's and little B's, or little A's and big B's together. And it should be 1/4, 1/4, 1/4, 1/4, depending on how meiosis is happening. All these ways are equally likely according to the law of independent assortment, because of the way these chromosomes line up independently of each other, and can be separated in these different random combinations during meiosis. Anyway, that's the very end now of these topic 3 here, these notes. You guys have lots of practice questions to go through in your notes, and there's answer keys posted, and we'll do a lot of this in class. I'll see you guys later.